Thanks for joining. I know it's after lunch, and I hope you're still active for this session. This is Karuna Nadadur. I'm from NVIDIA. I'm a solutions architect on AI and generative AI. Anything that is happening around the NVIDIA software stack, we kind of bring that to our partners and customers. I'm joined by Srijit. You want to introduce?
Hello, everybody. I'm Srijit. I'm a specialist solution architect at Databricks in life sciences. I specialize in data science, AI, and ML. I work with most of the biopharma customers in US and help them in their AI journey. So excited to be here, and hope you like the session.
Yeah, with that, let's get things started. So what are we going to cover today is, obviously, coming from NVIDIA, we are bringing the software to our customers and partners. And BioNeMo is one such special software we have created for a domain, which is the life sciences domain. And it primarily targets a lot of customers who are into drug discovery. That's why this is an important session if you're in that space. So what do we cover today is we want to set the context of why is it important and why you should take this seriously. The next is how does BioNeMo kind of work in the nutshell? What is it, and what is the difference between an open source model that is out there versus what BioNeMo brings to your table? And we'll get into the Databricks side of things, what are the key components that are being leveraged for this whole integration path. And then we'll talk into the architecture. The architecture is going to give you a perspective about how this was wholly implemented holistically, and then we'll get into questions, and then we'll get there. Okay. So what if you're a data scientist or a researcher who's getting into a problem statement like, how do I fold the protein, and how do I do a molecular analysis quickly, and I want to throw out multiple iterations of these in a fast pace of time? How do you do that? If you're caught in that whirlpool, you're in the right session. And then you want to just explore, scratch the surface, and I want to use a model that is already out there, and you want to go do the inference on your already existing input sequences, whether it's chemical or whether it's biological or molecular components. And all of your data could be sitting in a platform like Databricks already, Data Lakehouse. And how do you leverage the data for these kind of new inference or fine-tuning or training workloads? So in a way, you want to bring the workload to the data, and you don't want to be moving the data to the workload. And most importantly, all these experiments that you do is not going to be the end of it. You're going to continuously improve. You're going to keep your entire experiments in a location that you can go back and look at it. You want a platform to trust where you can actually go back and look at your results and then share it, and then finally go and deploy it in a place where you're successful. And most importantly, since I'm coming from NVIDIA, you must know all of these Gen AI or AI experimentation, it's only possible if you are using a heavy machine like a GPU, right? So GPU is a whole baseline for you to go and play around and use all of these experimentation. So you don't want to be caught in hey, how do I get the GPUs available for my experiment? Is it readily available? So Databricks already has these GPUs available for you. So you can go and pick and choose the GPU size, and for the workload, configure it and move on. So all of these are going to be answered as we move along in the session, and then we'll get into the specifics of how this can solve your end-to-end pipeline build easily, and how you can actually go on to do multiple iterations without having to scratch your head. How's it going to be end-to-end taken care, right? And then most of it, wherever your data is, where you're going to bring that workload to the data. So that's the most important thing. So if you're in the life sciences space, and if you're doing drug discovery, or if you're doing a chemical sequencing or a molecular analysis, if you're not using AI, you are already late into this game. AI is changing things, perspectives very fast, and these models that are already out there is going to make a whole lot of difference with your competition. So if you're into this space, and if you're not using AI, you should start thinking about it. So what is NVIDIA BioNeMo? Like, what is it? Like, it is nothing but a fully deployed solution, like a framework. It's a containerized framework for all things that you want to do, whether it's a protein folding, or whether you want to do chemical sequencing, or you want to do molecular analysis. It's all models that have been pre-trained, and for you to go and use them for your inference, or you want to do a fine-tuning, or you want to do a continuous learning, continuous pre-training, you can do all of that in one single giant container. So if you think about it, this is already out there pre-trained, and then for you to use on your platform of your choice. And then on the right-hand side, what you're looking at is NVIDIA has its own way of deploying all these BioNeMo frameworks. So we call it the BioNeMo NIMS, which is our inference engine, where the BioNeMo products or the BioNeMo models are available for you to use directly. So the framework is open source on the left-hand side. So it's available for anybody to go and use it. You just need the GPUs. And if you look at it, BioNeMo is primarily built to make sure that it's optimized for GPUs. And then when I say GPUs, like we have built it using transformer, we use the neural network in the back end. So make sure like you're getting the best performance for all these deployments, whether it's fine-tuning workloads or your inference workloads. So in a nutshell, like what is the difference, right? So all these models came as a white paper at some point, and then there is an original publication of all of this out there. And then why BioNeMo is like BioNeMo took all of that open source models, we have made it into an enterprise product. So what you get with this is high performance, and then the quality of the output. You get pre-created pre-processing, so all of this analysis cannot be done without preparing your data. So if you have to make all of this work, you need a lot of pre-processing, and we are giving you all of that curated already so that you can go and use that without having to spend a lot of time, how do I figure this out, right? Next, we also give you a lot of pre-configured YAMLs and the deployment end-to-end so that you don't need to worry about how do we go and deploy this, right? And finally, when you are ready to deploy, we are also offering a licensing support. Like the support model is out there for an enterprise level where you can actually go and take advantage of that, where if something goes wrong or you want to make sure like it's not, I mean, you know how the open source community works, right? So it's ever evolving, but there's not many people who can help you with any issues or problems. Like you need to be handheld at some point. We already talked about this, but going a little bit one step further, like I want to bring it to our attention, like all the models that are available. So on the right-hand side, what you're looking at is all the BioNeMo framework. It's fully Dockerized container, and it's available for free to use, and then you can use it for training or fine-tune. And then once you're fine-tuned, you can take that checkpoint and then use that for your experiments, or you can actually use that for your deployment, right? All of this RNA, DNA analysis, whether it's a protein folding exercise, any of this diff-dog that you're doing, all of these are available for you in the BioNeMo framework. But if you were to go and deploy it in the NVIDIA stack, we have something called BioNeMo NIMS, but this session is not about that. We're going to talk about how do we take this right-hand side and then go and deploy it on Databricks, where your enterprise data is sitting, right? So that's what this demo is going to be about, and that's what we want to convey to you, that, hey, we can integrate with partners, and then this is totally flexible how you leverage it in your production environment. And I want to go deep into this, but to prove a point, like why BioNeMo in terms of performance, like we have seen tremendous amount of inference throughput and latency taken care, because we have gone in and then tuned it for the GPUs, right? So if you're deploying GPUs for these kind of workloads, you want it to be the most optimized. And that's why our inference has been the fastest coming to this point. And what you get, I mean, this is the end-to-end workload exhaustive list of things that you can do, starting from pre-training. You want to continue to train your model, like take a foundation model, like let's say the ESM2NV, and then you can do a parameter efficient model, like a fully parameter efficient fine-tuning. You can also do a LoRa. You can build your own adapters where you take a checkpoint and then use it for different purposes. Like if you can purpose build an adapter, and then that adapter could do one task, whereas you can build another LoRa adapter that can go in for a different task. So you can pick and choose what you want to do with these BioNeMo models, because we are offering those LoRa adapters for it. What we are going to show today is the standard fine-tuning, like how you could take a foundation model from BioNeMo and then download it and then go and use your data sitting in Databricks, then go and fine-tune it from that point. And then if you look at it, like all the examples that you're looking at in the bottom, like a subcellular localization prediction or a melting temperature, all these sequence levels, classification or a regression or a token level classification, all these are examples we have curated in that notebook. And that's the notebook that is going to be available for you, and you can configure it the way you want it. With that, I will have Srijit talk about Databricks.
All right. Hello. So why Databricks is the platform? Because it's awesome. But honestly, like Databricks gives you all the nuts and bolts so that you, as a scientist, can do your work. Like you are not worried about configuring security, you're not worried about configuring GPUs, picking up which libraries works together and all those kind of things, right? We do that, we take care of all those stuff, right? Unity Catalog is one of the components in Databricks platform that takes care of end-to-end security and governance for all the artifacts that you can put in the lakehouse, right? It also tracks lineage. And then, because you are working on highly classified stuff, you want it to be protected, right? All are highly IP protected stuff. And all this governance comes out of the box from the platform. Now, you have your data in Databricks Lakehouse. You don't want to be like copying this data over like multiple times, because these genomics workloads, these data files are sometimes in terabytes, right? You don't want to be like keep copying this over and over again. So you can take advantage of these data sets that's already in Lakehouse, like from in place. Another cool feature in Databricks is we have a managed ML flow backbone, right? So for all your data science experiments, end-to-end model management, experiment tracking, everything is inbuilt into the platform. So you don't really have to do anything. You just need to keep logging your experiments, and it automatically tracks it for you. And then the compute that we have, you can pick and choose whichever VM you want, like GPU, CPU, and then you can pick which Databricks runtime you want to deploy, which means that Databricks runtime is nothing but like a set of curated libraries that we know works together, so you're not juggling with library management, right? One of the features in Databricks Compute is that you can run a pre-built Docker container on Databricks Compute. Now, that's the feature that we are taking advantage here, because all the beautiful things that NVIDIA has done and given it in a Docker container, we take it, sprinkle some pixie dust over it so that it works in Databricks platform, and we bring up the compute and attach a notebook. Everything that's available in BioNeMo is available to you in your familiar notebook-based environment. Right? How cool is that? Now, again, like Databricks in AWS, Azure, and GCP, so pretty much whatever you do, like to a great extent, is cloud agnostic, right? You can easily reuse and port it. Now, and you know that Spark can scale, right? So once you're done with your experiment, your notebook is all good. You're on hundreds of experiments, and you're good with one of the results. You can take that and scale it to like terabytes of data right from that notebook in Spark, and it'll run no problem, right? So this is why Databricks is the platform for running your genomics, your life sciences workload. So today we'll be taking advantage of few of the features, like the Unity Catalog, primarily for your assets, governance and access control. A Docker cluster with the NVIDIA compute, the ML flow for the experiment tracking, and a notebook-based development workflow, and we'll also show you how we can use that in a batch-based Databricks workflow, okay? All right, let's get into it.
Action time.
So, oops, sorry. All right, hope you can. Can you see my screen?
Thumbs up.
Is the size okay? Should I like zoom in a little bit? Let's zoom in anyways. All right. So what we are going to show you today is a fine-tuning exercise, right? We are going to take a model that's prepackaged in BioNeMo. We are going to fine-tune it using a regression head for this example, but you can do classification as well. And then we will take that weights, store it in Unity catalog, track that experiment in MLflow, and then use that weights for an inference on data that's in Lakehouse, okay? Before we do that, let me show you the pixie dust, right? So for people who are familiar with Docker, we have taken the Docker container that BioNeMo, so an NVIDIA team has given us, and these are all the pixie dust that we have put on top of it so that it works well with Databricks compute, right? It's just few layers of libraries that we added so that it can easily integrate with Databricks compute. Now, once we build this image and push it into any repository, like if you have your own container repository, you can use that as well. I have put it in a Docker Hub repository. So once the container is in the, once the image is in the repository, you can now bring up a Databricks cluster. So you can see here my great BioNeMo cluster, right? And if you go to the advanced options, you can see I'm using the BioNeMo Databricks build, which was built from the image that was provided by the NVIDIA team, right? Now the cluster is up and running. That means you can actually attach a notebook to it and take advantage of all these packages that NVIDIA provides. Now the choice of VM that you bring in is dependent on the model that you want to use. Here I'm using an A10 GPU. For bigger models like Evo, you might want to bring in like an A100 or an H100 GPU. But for this ESM, A10 seems to be sufficient. So let's see how we can fine-tune these, right? So like apart from all these libraries, some boilerplate stuff, I'm going to bring some data from a Databricks volume, right? And I don't know how many people are familiar with volumes, like Unity catalog volumes? Okay, cool. So Unity catalog volume is a feature in Unity catalog where you can store your unstructured data and fully manage it using Unity catalog, right? So assuming these data files are like H5AD files or FASTA files and all those kind of files, right? You can dump them in the Unity catalog and manage it. Here it is a CSV file. It's good old CSVs. I have taken a data set that's available openly in the internet. I think it is the E. coli data set. But that's not important. What we are trying to do is take the sequences that's in the data set and train a regression head on top of it, right? Now, these are some hyperparameters that are required by the BioNeMo container, so the BioNeMo model. I'm just going to copy over the data locally. For now, that's how it works. We are closely working with the NVIDIA team to have better integrations. We are going to expand this relationship further and working on better integrations where you can kick off these jobs at much more easy way. Then it's as simple as this. You load up an ESM to a model, just one line of code. Load ESM. So ESM2 comes in two flavors today in BioNeMo, a 650 million parameter and a 3 billion parameter. So I'm loading a 650 million parameter model, and it is sufficient for this exercise, right? So let's do some fine tuning. So before we do fine tuning, I'm just going to take a look at the data. So you can see the sequence, you can see the target, and I'm going to just ignore the remaining columns, right? And this is the data we are going to use to fine tune. And I prepared a separate validation data set so that while the fine tuning exercise is running, you can do some validation as well. finally, I also have an infer data set, which is basically the test data set that I can use to see how well the model is doing after fine-tuning. All right. Now, so the very first thing that we do, like if you're already using data science in Databricks, you know this, like we set up an ML flow experiment to track. Sorry about that. So we set up an ML flow experiment to track everything, right? So we start here. We set up an ML flow experiment here, right here. And I don't know if you noticed it, but we are installing two libraries, right? One is a PSUtil and PyNVML. What these libraries do is it enables the libraries, ML flow libraries, to grab the system metrics while the training runs. What that means is you get to know your GPU utilization. You get to know how your RAM is doing, like how your hardware is coping up, right, with this, with your experiment. The first thing you do is start a run and then log the parameters, because it's extremely important, because before a successful fine-tuning run, you might be running this like 100 times. And then after all these runs, you might be wondering, oh my God, yesterday I got a good result. What was the parameter I used? I forgot, right? But since you're logging these parameters to ML flow, all these runs are recorded. You can go back in time and then see which run worked best and what is the parameter that was used, right? And then we call the fine-tune API that was provided by NVIDIA here, right? So we pass in all the parameters. Today we are using just one GPU. And what we are doing here is, so while the fine-tuning run is happening, the BioNeMo packages spit out TensorBoard logs, events, right? So it tracks the loss, the validation loss, how it's progressing. So all those TensorBoard logs, we are reading it and logging into ML flow, right? So we capture the system metrics. Now we have the model metrics while the training is running, right? So then we are going to take the model checkpoint. So the fine-tuning is done. Now we want to test it out, right? So with the checkpoints locally, right now the fine-tuned weights are local to that cluster. With those local weights, I'm going to run an inference on the infer data frame that we have, right? So I run the inference, and then I can see I got it was a 50-row data set. I can see the regression output here. It also outputs the input IDs and the embedding, so you can take a look at that too. So you can see the labels and the predictions. Please don't judge me by the predictions, right? No, it's bad. It's just 10 steps, okay? It was just for demo. But you can see the training worked fine. It made some predictions. And now I can use ML flow to evaluate that prediction, right? So ML flow evaluate lets you log your evaluation metrics into the experiment, right? So you can have the data, the expected output, and the actual output, and it generates all these metrics. So now we can end the run, and we can take a look at that, right? Let's go to the experiments and take a look at the results. So you can see, so this is experiment. You can see I ran 3 times, 3 days ago. And when you open that experiment, you can see all the parameters that we used, all the system metrics, and the model metrics as the training was progressing, like how the loss was progressing as the training was going on. So this gives you an insight into how many epochs are needed. When should I stop? Am I doing too much? Am I using the GPU too much? I can also look at the system metrics now as to how the GPU was used, because you are paying for the GPU. You want to make sure the batch sizes are in a way that it takes maximum advantage of the GPU that you're bringing up, right? So you get to see the system metrics as to how the training ran. And then also you got the results. So you see this is the evaluation results, like the R2 score and the mean squared error and all those things, right? So everything is captured in this experiment, and every single run that you do will keep adding to it. So it is like a running log of all the runs that you did with those BioNeMo fine-tuning models, right? So now we are happy. We are good with the fine-tuned weights. Now we want to use these weights at scale. Like, let's say I have a 10 terabyte data set that I want to like run the inference on, right? What do? We take advantage of a Databricks platform feature called workflows, right? So what do? I create a new a notebook. This is a parameterized notebook. So this is a parameterized notebook where I can pass in parameters so that I can pass in different parameters whenever I need. So I pass in the catalog and the schema where the weights are stored, the location of the weights, where the data is, and what type of task it is, regression or classification. And then I also pass in the variant that needs. Okay? And with all these things, what I'm doing is I'm copying the weights over. So I can copy the weights over from the Unity catalog volume where we stored it. So what that means is once the fine tuning was done, I copied the weights over to Unity catalog volume so that when I bring the cluster down, I'm not losing those weights. Otherwise, it's in the ephemeral storage, right? So now I can use those weights, copy it back from the Unity catalog volume, and run the inference again, right? And now this time I'm running it as a batch job, so I can scale it up to how much how bigger the data set is. And I can see the inference job is running. And I can now copy the results back into another Unity catalog location, right? So what do? I create a workflow. So if I go to the workflows, you can see the inference job. So it's a simple workflow right now. It's just one notebook. But the compute that I'm going to bring up is the same compute that I used for fine-tuning, the same type of compute. I still use the same Docker container, and then bring up the compute. And then when you run it, you can see it successfully ran it. But now this is running as a background job. Like you can schedule it for like daily or weekly or monthly, however you want, right? So yeah, so that's the demo. Let's bring it back. All right. So how cool is that, right? So basically what we saw here is, just to wrap up, we brought in the image from NVIDIA, created our own little image with all the pixie dust on it, and used that to bring up an interactive cluster, right? Okay, can I point? Yeah, so we brought up an interactive cluster, used notebooks to run fine-tuning on that interactive cluster. The fine-tuning weights were stored in a Unity catalog location. And then we used those weights in a job cluster using those images from BioNeMo for NVIDIA and ran it as a batch job, right? And then the results goes back into Unity catalog. We are, like, as I told you, like, we are constantly working with the NVIDIA team. We have pretty good partnership going. We are looking at much better integrations to enhance the performance. We are also working on something like Genesis Workbench, which is kind of an internal app that we are trying to develop within Databricks, so that life scientists get a easy UI on top of it, and they don't have to worry about the boilerplate code and underlying platform setup and stuff like that. So stay tuned. More things coming, more exciting stuff. Thanks so much for being here, and it was a pleasure being here.